today representing Scailyte. And yeah, I just noticed that it's actually a nice balance of talks in this session, I think, because it touches on both from kind of sample preparation and the technology to here we're talking about biomarker discovery, and then the next talk I think is more on drug discovery. And then I also noticed it goes from kind of more close to the clinic to a small startup to a large pharma, so covering the entire value chain. But yeah, so now I'm going to talk about what our company Scailyte is doing in discovering next generation biomarkers through integrating single cell biology and artificial intelligence. So to motivate basically our mission, basically we start off by observing this problem that everyone in drug discovery will be familiar with, which is the R&D productivity gap. In the sense, you could summarize this by these kind of 90-50 problems that currently around 90% of clinical trials are actually failing. And of those, around 50% of failed trials fail because they cannot demonstrate clinical efficacy. On the other side, even among approved drugs, around 90% of those only really work in about 50% of patients. And we believe that one important way to try and close this gap on both sides is by having better and more actionable biomarkers. So our preferred data type of choice to discover this next generation of biomarkers is single cell data. So I think probably everyone in this audience appreciates that these data types are extremely rich and complex. We can profile up to thousands of genes from tens or hundreds of thousands of cells, depending on how you scale up the experiment. And what's really interesting to us is that we can work now directly from primary patient tissue samples. So PBMCs are obviously an easy one, but also tumor biopsies, anything that can be dissociated, and yeah, maybe even not dissociated if you move towards more spatial techniques. And the goal of any type of biomarker discovery study, of course, will be to take this complex data and find out from within all of these potential sources of information, which are the molecules and the cells that are actually responsible for the differences in an endpoint that you've observed. And that's what's going to give you your biomarker signature. So I just have an example of a single cell data set here, which is actually coming from CAR T-cell infusion products. So this is a public data set that we've reanalyzed, and I'll get into that in some more detail later on in the talk. But here the data is colored by sample. So each of the different colors you see represent CAR T cells that were derived from a different patient, because this is an autologous procedure. And already you can see that there are some challenges with this data set, right? So there are patient-specific clusters indicating a high level of interpatient heterogeneity. And there's also kind of a really large mixed cluster in the middle that could potentially be difficult to untangle and kind of annotate with different meaningful cell types or cell states. So in general, this data is obviously too complex for a human to find the relevant patterns manually, and that's why we apply machine learning to try and solve this problem. I don't want to go in too much in details of our platform, but we use a technique that builds on representation learning. And why we do this is to try and improve the predictive capability of the features in the data set. So an analogy for this is in natural language processing, where these techniques have been really highly developed. So if you take a set of words from a corpus, just a very simple example here, a naive way to represent those might be alphabetical. But that doesn't actually tell you that much about the meaning, the semantic meaning of those words. A representation learning approach tries to automatically learn by machine learning a more meaningful and interpretable representation of those words. So after applying that to this data set, you might have a clustering that looks something like this, where you have now, you can distinguish meaningful differences. Another example might be, some of you could be familiar with the AlphaFold algorithm in looking at protein folding, right? So this is also trying to do feature learning on these complex features of protein structures and try and predict something that's more meaningful. So what we try to learn at Scailyte is a cell representation. So from this large set of features that are measured in the single cell data, if we're talking about single cell RNA-seq, that would be transcripts, right, or aggregated to genes. We recognize that the raw data is not necessarily the most meaningful representation if you want to discover particular biomarkers or a way of separating patients or cell samples based on a particular endpoint. So using our approach, we can learn a new representation of the features in a new space that actually ideally would lead to a better separation for whatever your given task is at hand. So that could be predicting a diagnosis of a patient's disease, predicting stratifying patients based on response to treatment, etc. And just to compare a little bit our approach versus sort of classical single-cell analytics to show why we're different and what the advantages are. So the typical approach, if you're given a large data set like this, most people would start by clustering cells in an unsupervised manner. This can give you some information about the cellular neighborhoods and similarities, annotating cells by cell type, and then comparing the outcomes within clusters. So let's say cluster 1, you would compare for patient versus control, and et cetera, et cetera. So this can lead to some interesting insights, but it also has some potential drawbacks. In particular, the clustering step introduces a bias. So anyone who's actually done clustering on single-cell data knows that as a user, you need to set some parameters here, and this can alter what you get out of it. And indeed, this is a type of feature engineering. Cell type annotation also requires a pre-existing reference. So in that sense, you will only find what you know to already be there, or what you're looking for in the data. So it limits actually the capacity for de novo discoveries. And this approach of comparing outcomes between clusters actually reduces the sensitivity of the analysis because you've gone from single cells back to a cell type level. That means that if you have a really subtle signature in the data, you could miss it because it could be diluted by irrelevant cells that are in the same cluster, or your signature itself could be spread across multiple clusters. So with our representation learning approach, we reduce this bias because there's no clustering or cell annotation required up front. In fact, the algorithm learns directly and automatically which are the specific cells that are associated with the endpoints of interest. And we also, therefore, keep a high sensitivity on a single-cell level. So we keep the single-cell level throughout the entire analysis. We can go back and identify which were the specific cells in the original data set that were most predictive of that endpoint. This is just an overview slide of our platform and our workflow. It's just so you kind of know on a high level how we work. So our platform is called SkyVision. So we take as inputs single-cell data. This could be different data types, not only single-cell RNA-seq, but we also work with proteomics or multi-omics data types, and the endpoint of interest. We can also integrate other data types like clinical characteristics or other bulk measurements. Then we train a series of our machine learning networks, again, with the goal to find the patterns in the data that are most predictive of the specific endpoints. And of course, we always validate the predictive performance of those models based on a held-out test set of the original data set. And the output of this process is that you get two things. First, you get a model, which ideally will give a high predictive performance and therefore can be used then to predict endpoints on new samples coming in, which you might not know. But it's not a black box either, so we can open up the model and we can interrogate it and ask, what has it actually learned? What are the important features in terms of both molecular features and cellular features that it used to make its decision? And that's how we derive then the actual biomarkers. We also work on translating these biomarkers into simplified assays that could be used in clinical and IVD setups. And obviously these could have a variety of different applications, from disease diagnosis to patient stratification and even characterizing the mode of action of a new drug or compound. And now I'd like to show a specific use case, which is predicting a relevant clinical endpoint in the cell therapy space. So again, this is the data actually that I showed at the beginning, this CAR-T data set. So these were patients with diffuse large B-cell lymphoma that were treated with CAR-T products, and basically the treatment, the infusion product itself was recovered, what was left over in the bags, washed out, and subjected to single-cell RNA sequencing. The endpoint that we look at here is actually a neurotoxicity, which is a common adverse event and potentially quite serious adverse event with these types of treatments. So we wanted to predict from the treatment product itself, which of course is cells, which patients might develop this neurotoxicity. This data set had only 24 patients, so it's a bit limited in terms of size. But because our platform also integrates a data augmentation approach, we were actually able to perform training on this data. And we actually could train models that performed on this particular data set with 100% accuracy at predicting the outcome. So you see that summarized on the top. You have both the AUC that shows a 1.0 and the predictions of the held out samples. So we were correctly able to predict which patients would experience a high versus a low grade neurotoxicity. And then we also could identify, as I said, the exact cells and genes that were responsible for this prediction. And when we analyzed those, so we found cells in the CD4 and CD8 compartments. And we could find signatures looking at, for example, the enriched pathways and GO terms related to cytokine signaling, immune response, inflammation, things that suggest that we're actually picking up a relevant biological signal that could lead to these adverse events. And what's also interesting about this analysis, so this is public data, so we could compare our approach to what the authors did, actually, and see if we could find something more than what they found. So in their publication, they take the two standard approaches that I described. So they do cell type annotation, differential expression on annotated cell types, and also an unsupervised clustering approach. And they stated that they did not find any significant differences in the gene expression patterns between high and low grade toxicity in either the major CD4 or CD8 T cell compartments. On their clustering approach, they did identify a rare cell population that was associated with toxicity, which they called IACs. They're circled over on the left side of that TSNE. But you can see that this is kind of an outlier cluster. It's maybe a little bit suspect in the sense that it's only present in a subset of the patients. And even if it is not a technical artifact, it doesn't fully explain the observed toxicity. Whereas the cells that were automatically selected by SkyVision, actually, we did recover also these IACs, but we also recovered cell populations within both CD4 and CD8 compartments, as I mentioned before. So we were able to use that to actually characterize what are the differences in the T cells, which is the main, what you expect to be in the CAR T product. And also, when we quantified the proportion of cells selected by our algorithm in each sample, we found that this could actually itself perfectly stratify these patients by high versus low grade toxicity. So to summarize here, we show that we could both recapitulate what was found by the authors of the study and add some additional value that wasn't detected before in terms of potentially actionable biomarkers, which could be used for either screening or potentially even improving these therapies to reduce this type of toxicity. So that's all I have time for today, at this moment, to show you, but I would also hope that I've piqued your interest in what we can do, and invite you to come by our poster during the poster session tomorrow, where we have some more use cases, also a lot more technical details on our model implementation for those who are more technically minded. And we also have a booth, which is number 37, where we're more than happy to discuss how we can help you discover better biomarkers in your data. Thank you.
Thank you. It's obviously the important thing to do today, that you combine these machine learning AI things to omics data. I was wondering about these combinations to other types of data sets that you could enrich further if you have clinical data or let's say any other factors that you would like to engage with. In other session there was talk about the data lakes, and I guess this is still an issue, that how much we can bring additional qualities that will still further refine these details and kind of maybe identify further qualities on the data. So I don't know whether you have any comments on this.
So in terms of integrating other data types, we've already been working and have some examples where we've integrated clinical data together with single cell data in the models. And we can see some improvement when it's relevant, let's say. I think there's, always it's the question of, is it better just to have more data or to have better data? And I think it's important to ensure that the data is of good quality and that it's, actually relevant to the question at hand. But in general, that's another advantage, that we can really flexibly combine different data types, also different modalities of single cell data. On the protein level, for example, we have examples where we've done site-off analyses with multiple panels, and we can integrate them together and train one model that combines the two panels. So yeah, we definitely can do that. On the sort of data lake or data siloing question, I completely agree that this is a really important topic that a lot of people are addressing now. And yeah, I think we don't have a specific solution, but we see that that's a limitation, especially for training more machine learning models, the more data that's available, but also standardized and harmonized, then the more you can do. We find that often the limiting factor is actually the quality of the metadata that's available, rather than the genomics data itself.
Thank you for the talk, very interesting.
Thanks.
Can I have a slight clarification on the number of patients you mentioned being enough to power the analysis, because 24 patients in a single cell study is already relatively sizable given the expense to generate this kind of data. So, and you mentioned data augmentation to overcome some of that, but can you give some guidance as to at what point does this become powerful enough to be effective?
Yeah, so the cost is always an issue. That's one downside. But well, in terms of absolute numbers, it will depend, of course, on the strength of the signal, which you don't know up front. Our experience is that getting down to numbers around 2025 is kind of the minimum that is really reasonable to work with. For our own internal discovery projects, for example, we have an endometriosis project that's actually progressing now into IVD development. We've worked with cohorts of more like 60 or so initial discovery samples. But yeah, it's something in this range is I think feasible, and it's true that these data sets are sizable, but what I've seen, the trend in the last few years is that the size of data sets people are generating is growing. Anyway, this technology is becoming more accessible. I think it will only continue to, and hopefully the price will also drop if there's maybe more competition on the market. I think there's some companies here today that are bringing new technologies in single cell. So.
Thanks for the presentation. So you mentioned that, maybe I'm wrong, but your approach is deep learning based.
So it's derived from a convolutional neural network approach. I would not say it's deep because it's actually rather shallow, but the advantage of that is that it stays more explainable.
That's exactly the question. Like, how do you validate explainability compared to clustering methods
Well, to validate the explainability, I think you need to look at the biology eventually, right? So, I mean, we can do studies like the one I've shown where we compare what we find versus what is published and sort of validate in that sense. We can also work on synthetic data where we introduce perturbation or spike in synthetic cells and see if our algorithm recovers them. So we've done that also mainly with site-off data, but also some single-cell RNA-seq data. So I think those are the types of approaches that we can use to validate on the biological side.
Sorry, last question. And if the information wants to be used for clinical approaches, isn't there like an obstacle because machine learning or deep learning approaches are not yet ready, validatable for these purposes?
Well, I would challenge that a little bit because I think there are more and more, let's see, medical devices incorporating machine learning or AI that are being approved by the FDA. But yeah, okay, there are challenges for sure. And actually, our main approach now to go towards the clinic is to move from a complex signature discovered in single-cell data to something simplified that could be implemented in existing clinical infrastructure. So like a flow cytometry panel or targeted PCR or something like that.
That's going to be my question also, that, I mean, when you go to single cells, and then obviously it's if you think about tumor as an example, so there's basically lots of different cell types, and which are then either part of the biology or somewhat contributing to the tumor development. So then and with respect to this clinical thing, because there's lots of need obviously for biomarkers to be validated and translated to patient diagnostics. So then, how to speed up this? And obviously it's still somewhat costly and slow process to make all these things. Can this be automated somehow? And then just to advertise Finland a little bit, so with regards to these other types of data lakes, so we have now the FinData, which is a national cluster that has 33 national electronic records on patient aspects that are then can be actually crossed to this data. So that would be an offering more, let's say, accredited plus also kind of GDPR compatible data to compare for this data. But there's kind of two things. One is this, how to really speed up this translation to clinical, what was pointed out.
Yeah, that's a really good question. I don't know if I have an answer off the top of my head how to speed it up. I think better collaborations, more openness in data sharing. I think a lot of, we have so many tools that are available to us now, but seems like often what stands in the way is more, coming to agreements between different parties to work together. But yeah, I think also having the infrastructure in place in clinical trials, we start to see that companies, pharmas developing drugs are starting to already plan single-cell experiments in their clinical trials. So it's a main data that they're capturing, but this is pretty recent. But I think if that progresses, then we can build off of that to sort of progress more quickly and hopefully also in a loop be able to intelligently and better design clinical trials to become more efficient. Right? So that's, the point about the data lake, I think I lost a little bit along the way. I'm sorry, but we can maybe follow up on it later. Yeah.
Very good.
Okay, thank you.
Thank you all. Unfortunately, we're missing Tobias.